Welcome back. I hope you had a nice break and you had a bit of a chance to make a new cup of tea. I got assaulted by this probiotic up here, which is a giant Lactobacillus acidophilus, bought for me by my son many years ago when we were in the United States. And so I thought we would do the second half of this lecture with this giant Lactobacillus on the table, because that makes everything better, right? Yes, I think so too. Now, imagine that a consumer comes into your pharmacy and they say, okay, thank you for explaining which products are on the market, but which one of these products should I actually buy? Because it's going to cost me money, isn't it? So if I'm going to spend money on a probiotic product, which product should I buy? Because I don't want to waste my money. Now, that's a very good question. It's one that we asked ourselves a few years ago. And so in order to answer this question, we did what any sensible academic would do, and we got an MPharm student to undertake it as part of their research project. So the first part of the data I'm going to show you in this part of the lecture is actually recorded by an MPharm student a few years ago who we sent across the road to go and buy some probiotic products. I think she went to Waitrose, Boots and Holland & Barrett and bought as many probiotic products as she could. And the reason that we did that is because we simply wanted to test them to see how efficacious they were. Okay? And in that way, we could then advise a patient, if you want to buy this particular type of product, it should be effective. Maybe if you buy this particular type of probiotic, it might be less effective. So it was really just to explore how we could test probiotic products and then give some sort of guidance as healthcare professionals to members of the public. Okay. To do this, we asked a number of questions that we thought needed answers in order to provide a sensible basis for comparing probiotics. Okay, the questions that we asked were: did the probiotic product arrive in a viable state? So arrive in a viable state means, does the probiotic product contain live bacteria, or at least bacteria that you can recover to be alive, of the strains that it says on the packet and in the numbers that it says on the packet? So that was the first question. Did they survive? And when I say survive, I mean the challenges of oral delivery. So that means patient swallows the product, and that product is going to go through stomach acid. So the bacteria have got to survive being in a relatively harsh environment, haven't they? So could they survive stomach acid? And then could they thrive? Really, the thrive, we chose that word because it rhymes with arrive and survive. Okay, so could they thrive? In other words, once they'd been exposed to stomach acid, could they actually grow and proliferate in the gut? Because if they can't, they're not going to do anything, are they? So they've got to arrive in a viable state, they've got to survive the challenges of oral delivery, and then they've got to thrive in the gut. And if they can do all of those things, then it should be relatively effective, right? So that was the set of questions that we got the student to ask. How did we do that? We made her go into the laboratory and run a number of tests. The first one was arrive, was simply a matter of rehydrating some of the probiotic products if they were solids, or plating out the liquids and seeing how many bacteria were there. So you simply plate onto agar, I'm sure you're aware of that. You leave them in the incubator and then you count 48 days later, so 48 hours later. And so she simply enumerated how many bacteria were in each of the products. That was relatively simple. The second one, which was the survive, we developed what we called a tolerance test, which is a fancy way of saying we created a model stomach with artificial stomach acid, and then we dunked the bacteria in the acid for various periods of time. I think we went up to 30 minutes exposure to stomach acid, and then we plated out again to see how many of the bacteria were able to cope with sitting in acid for that long. And the last one, because we can't really do in vivo growth in the gut to see how the bacteria actually colonise, for all the reasons I mentioned before the tea break, we did an in vitro test of how the bacteria grew. And we did that with the technique that I use in my laboratory, which is called isothermal microcalorimetry. But, as I alluded to at the start of this lecture, I fully recognise that this lecture series is overwhelmingly complex, and I don't want to make it any more complex. And so I'm going to leave the microcalorimetry section out. And if you're interested in understanding what that does, fire me an email. I'll talk to you about it separately. Okay? So, the first part is easy. When the student enumerated, which is a fancy way of saying counted, all of the bacteria in the products. What did she find? She found that all of the products on sale in the UK contained the right species as listed on the packet and the number as listed on the packet, which is probably unsurprising, isn't it? We have very strict rules about product labelling in this country, and I think it should come as no surprise that if a box for a product says it contains X, it did actually contain X. So that's very comforting. So there's no real data to show you for that, because in our testing, all of the products contain equal to or more than label claims. That's fine. The gastric tolerance testing, on the other hand, was kind of interesting. So she spent quite a long time developing a model stomach. It's shown on the screen in front of you. It's a glass beaker with 0.1 molar hydrochloric acid in. Really not very complicated, actually. So we just we took that hydrochloric acid, we added the probiotics, and we added them in the dose as specified on the bottle. So if the bottle says swallow the whole bottle, we added the whole bottle. Okay. If it says pour the sachet into a glass of water, we poured the sachet into a glass of water. We did exactly what it said on the label, and then we added the probiotic products to the acid and let it sit for various time points. And then we took an aliquot of the bacteria out, plated and enumerated. So the idea is how many bacteria were remaining relative to what was in the product at the start. Okay. The results, I think, were kind of interesting. They're shown on the screen in front of you. The graph looks a little bit busy, and I'm sorry about that. But on the y-axis, you've got the number of viable cells. That's the number of cells that grew when we plated out as a function of exposure time to acid. 0 is how many bacteria were in the product initially. 30 minutes was the longest we left the product in acid. So as you can see on the screen, the number generally reduces. This is not a surprise, is it? None of the probiotic bacteria we tested grow in stomach acid. But it was very clear that some products survived a lot better in stomach acid than others. So in the blue box at the top of the screen were the 3 best performing products. They were 2 liquids and 1 solid product. If you go and read the paper, it's the references there, Beneficial Microbes. It's open access, so free to download. We actually named the products, but I got in all sorts of trouble with the legal departments of the manufacturers for naming the products in this paper, and so now I just refer to them as liquid and solid products, and you have to go back to the original paper to work out which one is which. But those 3 products perform pretty well. Okay, they started at around 7 to 8, 10 to the 7 to 8 bacteria per mil in the original product. And after exposure to acid, even up to 30 minutes, they really didn't lose that many viable cells. And that's what you want. You want to swallow something, and when it empties from the stomach, you want that number of viable bacteria emptying into your gut. And so those products are pretty good. And what we found was, generally, and it's a bit general this, a liquid product performed better than a solid product. So I think you can see that in my grey box, 2 liquids and 1 solid. There's another liquid product which was generally okay for around 10 to 15 minutes, and then it started to lose viability. But in the main, the solid products, within 5 to 10 minutes, everything was almost dead. Okay, so what that means is sometimes the newspaper or various media people, they ask me for a quote about probiotics and what sort of probiotics people should buy, and I usually say I think it's better to buy a liquid product than a solid product, and this is the reason why. When we tested solid products, generally they don't survive exposure to acid very well. Now, if you're a manufacturer of a solid product, you're going to make some sort of argument as to why your product is okay. Maybe it's enterically coated. Maybe my experiment is completely rubbish. It doesn't matter what it is, but I make my judgment based on the data that I've recorded. And so in this instance, I think liquid products are probably better than solid products. Why is that? I don't hear you ask. And the answer is because think about a bacterium. If it's alive, it contains a lot of water, same as you and I. And so if you've got a liquid product, the bacterium is already hydrated. It's not trying to suck in water to rehydrate itself. If you've got a solid product, and by that I mean sachets as well as tablets and capsules, the bacteria have generally been freeze-dried to make that product in the first place. Freeze drying, as I'm sure you remember from the drying lecture, means the removal of water by sublimation. So you freeze the product, you place it under vacuum, you remove the water by sublimation. So the bacteria have been properly desiccated. And so if you swallow that, and it's sitting in stomach acid, the bacteria is having to imbibe a lot of water to rehydrate itself. And in doing that, it's also going to suck in acid. And it should come as no surprise that really a lot of the bacteria die as a result of that, because of the strong acid that they're trying to imbibe. Yeah. From all of our testing, there was one product that came out really quite well. It is one of the liquid products, unsurprisingly, on the screen before. And that product was Simproof. You may have not have heard of Simproof, and I hadn't heard of it either, but it happened to be in the news when we were doing this testing originally, because it had been used in a clinical study at King's College for IBS and IBD, and it was showing really positive clinical results. And so when we did our testing, it turned out that it was the one that survived stomach acid exposure the best. And so we thought, that's interesting. So unusually for me, I simply got on the phone and rang them up and said, I've been testing your product and it was really good in our in vitro testing. Can we come and talk to you about it? So we did. So we went down and talked to them, and since then we have spent a long time trying to understand what it is about that product which makes it effective. And I think some of the things that make it effective apply to all probiotic products. So I thought what I'd do in the last part of the lecture is I will show you some of the in vitro testing that we've been doing with this product, because it gives you a general mechanism by which probiotics work. And I think this general mechanism applies to all probiotics, but you just have to apply that, recognising that the formulation has to get the bacteria into the gut. As long as your formulation protects the bacteria from stomach acid and allows them to reach the gut alive, then I think all probiotics are going to work this way. Okay. Now, I should also say that there are quite a lot of data, and I'm going to whiz through it a little bit quickly, but you can take your time just to look at the data that are being shown in front of you. Don't panic. In an exam type question, I'm not going to ask you to reproduce these graphs. I'm simply showing you the data that we recorded, because it allows you to see where we get to when it comes to saying this is how we think probiotics are working. Okay? And again, any questions, just fire me an email and I'll do my best to answer them one-on-one. What is Simproof? It's a tremendous product. It contains 4 probiotics listed on the screen. So 3 of them are lactobacilli, so Plantarum, Rhamnosus and Acidophilus, and it's got an Enterococcus as well. It is made in a fermented barley medium. So you take barley, you allow it to sprout, which is called malting. You then dry, very much like making beer actually. You then crush that up, remove the husk to create a wort, and then you grow the bacteria in that wort. And so it's a fermented barley extract. You can see a bottle of it on the screen. We did some very interesting in vitro testing. We did not do in vivo testing for all the reasons I mentioned earlier. It's really difficult to do proper analytical science inside someone's gut. Okay, so we did in vitro testing, but we chose to work with a company that has the most advanced in vitro model of the human gut that we could find. So it's a Belgian company called Probiotics, and it has this really complicated looking system. It's called the Simulator of the Human Intestinal Microbial Ecosystem. Now I had to read that off the screen because it's so complicated, or SHIME. So it's called the SHIME system, and essentially it's a series of glass jars, 5 in sequence. There's more shown on the screen because it's running multiple tests. So 5 glass jars, and they represent various stages along the GI tract. The first jar represents stomach acid, very similar to how we did our own testing. The second and third jars represent the small intestine, and the fourth and the fifth jars represent the large intestine. And when they do the test for real, they populate the colonic jars, that's 4 and 5, with stool samples from donors. So the reason that some of those jars look a little bit brown on the screen is because they are actually a fecal slurry from human donors, and they allow that fecal slurry to sit and stabilise for a period of 2 weeks before they do the testing. So there's all sorts of bacteria growing in there, and they should be representative of the bacteria growing in the gut. Okay? Then the way the test works is you put your product at one end. Obviously, it's going orally, so it's going to go into vessel number 1 first, which is stomach acid. It sits there for 45 minutes. Then it empties into the small intestine. It spends another couple of hours going through the small intestine, and then it arrives in the 2 colonic chambers. Okay? The 2 colonic chambers represent both the near and the far, or proximal and distal regions of the colon. So when we look at the data in a minute, you'll see it says proximal and distal. So that means near and far regions of the colon. And the other thing they do in this system, which is kind of clever, is that they have a mucus. So they take some beads, they coat those beads with a mucus layer, and they suspend those beads in the colonic medium. So they can also remove those beads at the end, and so they can ask questions about where did the bacteria go. Did they remain in the liquid content of the colon, or did it go into the mucus? Okay, so if it's in the liquid, it's called luminal, and if it's in the mucus, it's called mucosal. So when we look at the data in just a second, you'll see it says proximal and distal, and luminal and mucosal. That's where that's coming from. Near and far, liquid mucus. Okay, once you've got all that information in your head, you should be good to understand the data that are coming up on the screen. So on the screen in front of you are 4 graphs. The 4 graphs are for the 4 species of bacteria in the product. I know there's a lot of lines on these graphs, but bear with me, it will be okay. And there is a little diagram in the top corner to show you what you're looking at. Grey means lumen of the proximal colon. Red means the lumen of the distal colon. Blue means mucosal layer in the proximal colon. Green means mucosal layer in the distal colon. So you can start to ask questions about where did the probiotics go when they were entered into the system. Okay. If you look at the acidophilus first, that's the top left, you might say to me, well, hang on a minute, Simon, there's no lines on that graph except for 2, which is a bit weird. What's going on there? And the answer is that Lactobacillus acidophilus, at least in simproof, isn't there in very high concentration, and so it's not really a probiotic in the system. It was there. It's helping them make the product. It allows one of the bacteria to grow during manufacture, but actually it's not there in high quantity, and that was reflected in the test. We couldn't really detect it in any of the regions in the in vitro test. So that's why there's not a lot on that graph. But if you look at plantarum, now you can see that the bacteria have started to go somewhere. You might say to me, what does it mean on the x-axis when it says C, T1, T2 and T3? And the answer is C is the control. So that means the microbiotas were allowed to rest for 3 weeks, but nothing was added to them. And that was the initial concentrations were measured. So effectively it's the time 0, initial concentration, control point. T1, T2 and T3, the system was dosed with simproof on a daily basis for 3 weeks, but measurements were only made after 1 week, 2 weeks and 3 weeks. So T1 means treatment for 1 week, T2 is treatment for 2 weeks, T3 is treatment for 3 weeks. So you can see what the change is as you take the product for a number of weeks. So if you look at the plantarum first, you can see that there was some plantarum in the control. So in other words, it's a natively existing species. But you can see that after 1, 2 and 3 weeks dosing with simproof, the numbers have massively increased, hugely increased, because that's a log scale. So they've gone from under 10 to the 2 to 10 to the 8. That's a huge increase in the number of plantarum. You can also see as you go across that graph that plantarum was detected in the proximal and distal colon luminally and the proximal and distal colons mucosally. So in other words, that particular probiotic managed to colonize and grow in all the regions of the colon, near and far, luminal and mucosal. So that's good, which is a sign it's a good probiotic species in the first place, right? Bottom left shows you the same data for rhamnosus. I think you can see, I hope if you're following. If not, stop, rewind, play again. That the bacteria managed to grow in the luminal contents of the proximal and distal colon. It did manage to get the mucosal layer of the proximal colon, but not in the mucosal layer in the distal colon. And the way I like to rationalise this is that obviously when the bacteria arrive in the colon, they're arriving at the proximal colon, and the first place they're going to grow is the lumen. It may be possible for them to colonise the mucus, or they may transit along the colon and get to the distal colon, lumen. And once they're there, they may also manage to get into the mucosal layer. So really the distal mucosal layer is the hardest area for the bacteria to populate. Then it's the proximal mucosal layer, and then it's the luminal layer, which is what we see here. So the bacteria grew in the lumen. They managed to infiltrate the mucosal layer in the proximal colon, but not in the distal colon. And then Enterococcus faecium, you can see, went everywhere. But the interesting thing about Enterococcus faecium is it's a native species in most people's gut. It's already there in massive number. So you can see the control, which is the first column, it was already there at more than 10 to the 7, and it went up to 10 to the 8, but it was already there a lot. So it's unsurprising that it was found everywhere because it's a typical species from your gut. And in fact, the reason it's called Enterococcus faecium is because it was first identified from human feces. That's where its name comes from. Okay. So really what that says to you is Lactobacillus plantarum and rhamnosus were very good. If they arrived in the colon, they generally managed to thrive, especially in the lumen, but also in the mucosal layer, which is what you want for an effective probiotic. The next thing, and there's a new set of graphs on the screen, is a little bit complicated, and I said already, most probiotic bacteria are lactic acid bacteria. They're called that because when they metabolize, they produce lactic acid as a waste product. And lactic acid is absolutely key to the mechanism by which probiotics are working, as I'm going to come back to in a little bit. And we've done lots of work looking at what lactic acid does in the gut. Okay, but right now this is simply showing you the concentration of lactic acid as a function of time when simproof was dosed onto the system. So that's top left, shows you lactic acid. The control shows you how much lactic acid was there initially, and then you can see the blue and the red bars show you how much lactic acid was being produced as a function of dosing with simproof over 3 weeks, and you can see a massive increase after week 1, which increased after week 2 and increased again after week 3, which is unsurprising, isn't it? Because they're lactic acid bacteria and they should be producing lactic acid. Now, what you might not know about the gut bacteria is that lactic acid is an interesting molecule because some of your gut bacteria, they use it as a food substance. Okay, so lactic acid is a C4 carbon, 4-carbon chain, and so some bacteria can actually degrade that and get useful energy from it. Some bacteria, when they degrade lactic acid, can produce propionate, which is a 3-carbon chain. Some can produce acetate, which is a 2-carbon chain. Some bacteria can use propionate and produce acetate, and some bacteria can produce butyrate, which is also a 4-carbon chain, but it's not the same structure as lactate. Acetate, propionate and butyrate are known as short-chain fatty acids, SCFAs. You may have heard of those because you can measure the short-chain fatty acid production in the gut, and generally short-chain fatty acid production is linked to a healthy gut. The more short-chain fatty acids there are, the better your overall gut health is assumed to be. Interestingly, if you look at the data, top right shows you how much acetate was produced after treatment with simproof, and you'll see not a lot of change. You might say to me, well, hold on a minute, you've given simproof 3 weeks here and nothing's changed in the acetate. And if you look at the bottom left, you'll say the same thing to me with the propionate. And I would say, yes, that is absolutely true. But what you've got to remember here is this is a cascade of events. The probiotic produces lactic acid. Lactic acid is a food substance for the commensal bacteria that eat it. Some of those bacteria produce acetate, some produce propionate, but there are other bacteria that utilise acetate and propionate as food substances. So you get this complicated cross-feeding interaction going on, and you don't see a build-up of acetate and propionate either in this in vitro test or in vivo, actually, because all of those molecules are being used up by other bacterial groups. Okay, so what you end up doing is producing lactic acid, which is kind of stimulating the production of all these other commensal bacterial groups through acetate and propionate production. The only thing you see an increase in is butyrate, which is shown on the bottom right, and the reason is because the commensal gut bacteria don't actually use butyrate itself as a food substance. This is an in vitro test, which means everything's being done in a glass beaker. There's nowhere else for the butyrate to go, and so it generally rises in concentration because it's not being used by anything else. In vivo, so in you, butyrate is a really important molecule. It's a molecule which is absorbed by your colonocytes, that's the cells lining the colon, and it's an energy substance for your own cells. So in the body, if your commensal bacteria are producing butyrate, that butyrate is being absorbed by the cells lining your gut wall and they use it as an energy source. So again, increased levels of butyrate are usually associated with good gut health. And so you can see in this instance, we dosed the system with the probiotic. That produced lactic acid. The lactic acid stimulates commensal gut bacterial growth, and we see an increase in butyrate production. And we come back at the end to what this all means for a mechanism of how the probiotics are working. There are some compounds that you don't want in your gut. In other words, they are negative indicators or indicators of bad gut health. Some of those are branched chain fatty acids, which I've called BCFAs, and another one is ammonium. So again, if you look at the screen, branched chain fatty acids on the left, you can see a reduction in production after giving simproof. And ammonium is more or less the same, a slight reduction in the distal colon, but no increase, which is really good. This graph is really complicated, and so what we did was we took samples from the gut and we analysed all of the different bacteria that are there, all 6,000 to 10,000 of them, but we're simply plotting here the phyla. So I mentioned phyla right at the start. They are the main family groups. So there are 6 shown: actinobacteria, bacteroids, Bacteroidetes, firmicutes, proteobacteria, the ones I talked about earlier, but there are 2 others that are sometimes seen, synergistetes and verrucomicrobia. So it's a little bit complicated, but on the screen, C means control, T means treatment, but it's treatment after 3 weeks. PC means proximal colon, DC means distal colon, and then we use stool samples from 3 different donors, so donor 1, donor 2, donor 3. So really the way to read this graph is, if you look at the column, which has a C at the bottom, that's the proportion of phyla in each donor at the start of the experiment, and the column to its right, which is the T, shows you the proportion of phyla after 3 weeks of dosing the simproof. Okay, so just compare each column with a C with the column immediately to its right, and that will show you how the phyla are changing after 3 weeks dosing with simproof. I think you can see that the proportion of phyla is changing in all donors after treatment with simproof. And this is one of the reasons why we think dosing with probiotics is so important for improving various disease states. Because we know that some of these disease states are linked with a degree of dysbiosis in the gut, and these data are suggesting that by taking a probiotic on a regular basis, you start to change the proportion of phyla in the gut. As long as you change them in a direction which is generally associated with good health, not with bad health, you start to alleviate some of the other conditions. And so we've done clinical studies in a number of disease states, including liver cirrhosis, Parkinson's disease, and ulcerative colitis. And I can share some of those papers with you if you like afterwards. We see the same effect in the distal colon, so it's the same measurement. It's just that the proportions of bacteria are slightly different as you move further down the gut. And we made a number of other measurements in this system. One thing we can do is we can measure how the cells lining the gut wall react to the presence of the probiotic. I'm sure you know this, but the cells in the gut wall can release compounds which are either markers of inflammation or markers of reduced inflammation. So you can have anti-inflammatory responses and inflammatory responses. Okay, so what you're looking for good gut health at least, is an increase in the production of anti-inflammatory compounds and a decrease in the production of inflammatory compounds. So on the screen in front of you are shown the 4 anti-inflammatory compounds that we looked for in the study, and you can see that in all cases their production was increased with the probiotic. And on this screen you can see the inflammatory markers, which in all cases decreased after probiotic therapy. So again, all of the data we're looking at so far suggest dosing with a probiotic for 3 weeks is improving general markers of gut health. All of this means we can bring together a mechanism for how probiotics actually work in the gut. You remember that I said most probiotic bacteria are lactic acid bacteria, and so they produce lactic acid as they colonise and grow in the gut, and that is absolutely key. One thing that the previous data didn't show you is that we've done a lot of measurements on how lactic acid affects opportunistic pathogens, so the types of bacteria you don't want in your gut. And our measurements show that most opportunistic pathogens are very intolerant to low pH. So as the probiotics grow and they produce lactic acid, they drop the local pH, and that tends to be very detrimental to any infectious agents that you've got, and it can clear up the infection without having to take antibiotics. Obviously, if nothing else happened, your gut would then be a lower pH than would be physiologically normal. So something has to happen to make sure that doesn't stay that way. And what happens is your commensal bacteria view the lactic acid as a food source. And so they utilise that lactic acid to grow themselves. The lactic acid goes away, and so the pH rises, and the bacteria themselves produce a number of short-chain fatty acids, acetate, propionate and butyrate. The acetate and propionate are themselves foodstuffs for other commensal bacteria, so they grow in response, and the butyrate is a fuel source for your colonocytes, and high levels of butyrate are implicated in good human health. So that's the basic mechanism by which I think probiotics exert their effect in vivo. So, we're at the end. You'll be pleased to know, not too bad. On the screen are a list of the main points that I think you need to remember when reviewing this class. The first one is that the microbiota in your gut is absolutely key to good health. If you have a dysbiosis in your gut microbiota, then it's linked to all sorts of different disease states, some you might have thought of and some that you haven't. Fecal transplant is a large stage resort after antibiotic therapy, but I think that probiotic treatment is a really good alternative, not only to fecal transplant but also to antibiotic use actually. It just needs proper delivery of the probiotics in the first place, and it also needs some good underpinning science to convince the medical profession that this is the way forward, and that's really what we're trying to do in our own labs. If you talk to a patient about probiotics, remember probiotics are classified as foodstuffs, so they don't need a prescription, and a patient can incorporate them into their diet on a daily basis. And finally, although we're not there yet, remember that the US FDA does at least recognise that next generation probiotics and live biotherapeutic products may well be available in the future. So with that, hope you enjoyed it, and I'll see you again soon.